Namaste Vidya, myself JVN Neha Saini from Physiotherapy and Diagnostics. Here we discuss about the anatomy of the blood vessels. There are three types of blood vessels: arteries, veins, and capillaries. Arteries are the vessels that transport the blood away from the heart. Veins return blood back to the heart. Capillaries, these are the microscopic blood vessels that allow exchanges between blood and the tissues. So this is the formation of capillaries, how these are formed from the arteries. The arteries receives the blood from the heart, transfer it to the capillary. Capillary carries the blood to and from the body cells. After exchange, the venous blood is transferred back to the heart. The arteries divide to form arterioles. Arterioles again divide to form capillaries. Capillaries at the venous end unite to form the venules, and the venules unite to form the veins. This is the total length of the arteries, or we can say the vessels, in the human body. It is more than 1 lakh kilometers. This is how the blood flow through the tissues. From the heart, the blood flows into the aorta, then to the arteries, then it is transferred to the arterioles and capillaries. After exchange, the venous blood is collected by the venules, and the venules transfer it to the veins. These veins collect and drains the venous blood into the larger vein, that is your superior and the inferior vena cava, which opens into the heart. So the arteries, they transfer the blood away from the heart. These are strong and elastic. They conduct the blood to the capillaries. They have sphincters. Capillaries, they exchange with the cells. Veins, they return the blood to the heart, and they possess the walls. This is the aorta, which divides to form the larger arteries and the smaller arteries. Smaller arteries divide to form the arteriole, which bifurcates or trifurcates to form the capillaries. After exchange, the venous blood is collected by the venules. The venules converge to form the vein. The venous blood from the vein is transferred to the vena cava. We have two types of circulation, systemic and pulmonary. Systemic circulation delivers the blood to all the body cells and carries away the waste. Pulmonary circulation eliminates the carbon dioxide and oxygenates the blood. This is the pulmonary circulation, which is on the right side. On the right ventricle, the venous blood is transferred to the pulmonary trunk. From here, the blood is transferred to the right and the left pulmonary arteries, then to the arterioles and capillaries. In capillaries, diffusion occurs, and after diffusion, the pure blood is collected by the venules, which is transferred to the veins and then to the pulmonary veins. These pulmonary veins transfer the pure blood to the left atrium of the heart. This is the systemic circulation. Systemic circulation carries the oxygenated blood away from the heart to the body and returns the deoxygenated blood to the heart. Now the blood vessels, since our human body having the blood which is transferred in your vessels, that's why we have the closed system of circulatory system, and these are containing your arteries, arterioles, capillaries, venules, and veins. Now the general structure of the blood vessels includes the three tunics. Either it is a artery or a vein. There are three basic layers. The internal layer is the tunica interna or intima, the middle layer is tunica media, and the external layer is tunica externa. Capillaries are composed of endothelium only. So these are the layers. The basic anatomy of the blood vessels includes the tunica interna. This is the first layer, or it is the innermost layer. The endothelial layer that lines the lumen of all the vessels, and in vessels larger than 1 mm, a subendothelial connective tissue basement membrane is also present. Then we have the tunica media, which is made up of smooth muscle and elastic fibers. These are regulated by the sympathetic nervous system and controls the vasoconstriction and vasodilatation of the vessels. Then we have tunica externa. It is also known as tunica adventitia. It is made up of collagen fibers which protect and reinforce the vessels. The larger vessels contain vasa vasorum which supply the tunica externa. This is the basic structure of a vessel, either it is an artery or a vein. We have the three basic layers: tunica interna, which is again made up of three layers, that is the endothelial and the subendothelial layer, and the internal elastic lamina. This internal elastic lamina, made up of elastic fibers, separates the tunica interna and media. Tunica media and tunica externa, they are separated by external elastic lamina. In case of veins, the endothelium folds to form the valves. Valves prevents the backflow of the blood. They are for maintenance of unidirectional flow of blood. This is the 3D diagram of the artery. Here we can see the three layers of the artery with the lumen. This is the histological image of an artery. Here we can see the endothelial layer, and beside that we can see the muscle layer, that is the tunica media, and the outermost layer, that is the connective tissue layer, the tunica externa. Now the types of arteries. First we have the elastic arteries. These are thick wall arteries near the heart. It includes the aorta and its major branches, and they have the larger lumen which allows the low resistance conduction of the blood. It contains lots of elastin, that is why it is named as elastic artery, which are present in all the three tunics. These elastic fibers enables the walls to stretch and to recoil to propel the blood. They withstand and they regulate the large blood pressure fluctuations. This is the pictographical image of the elastic artery. Here we can observe there are numerous elastic fibers in the three layers of the elastic artery. This is the histological image of an elastic artery. The appearance of zigzag structures is your elastic fibers. Then second type of the artery is the muscular artery. It is also known as distributing arteries. These are also known as medium-sized arteries. And the tunica media is having more smooth muscles and less elastic fibers, which are the major area of vasoconstriction and dilatation. They regulate the blood flow. Then the third type is the arteriole. Arterioles have diameter of 0.3 millimeter or less. These are the smallest arteries. They lead to capillary beds. Close to capillaries, single layer of muscles spiraling around the endothelial lining, and they regulate the blood flow to the capillary. In the diagram, we can see the structure of the arteriole. It is possessing all the three tunics of the blood vessels, but the tunica media is very thin. Then this is again the basic structure of the blood vessels. Now the venous system. The veins are formed when capillary beds unite. There are two kinds of venules: post-capillary venules. These are the smallest venules and they form when several capillaries unite. The walls are very porous, serve as the site for WBC emigration. And we have the second type, that is your muscular venules, which have 1 to 2 layers of smooth muscles, and there is no more exchange with the interstitial fluid. The veins, these are formed when venules converge, and they are composed of again the three tunics with an thin tunica intima, media, and there is thick tunica externa consisting of collagen fibers and elastic networks. Capacitance vessels, we can say the blood reservoir, and they contain 65% of the blood supply. That is why they are named as blood reservoir. So in the image we can see the three layers of the veins. The innermost is the endothelium resting on the basement membrane. Then we have the tunica media containing your smooth muscle and elastic fibers, and the outer coat, that is your tunica externa. In veins, the endothelial is again infolds to form the walls. So here we can see the superficial veins. This is the journal diagram to show you the structure of the veins. Now the venous walls. Venous walls are the infoldings of endothelial layer, and the valves present for unidirectional blood flow. In the diagram, we can see the venous valves. The venous flow is occurred by the muscle contraction. The diagram is indicating the venous flow. The unidirectional flow in the veins is assisted by the muscular contraction. As the muscle contracts, it causes the blood in the vicinity to be pushed up, and it is assisted also by the valves. The valves, after passing the blood from one point to the another, they become closed, and this prevents the backflow of the blood, and there is larger diameter in the veins through which more and more blood can be transferred. So this is the general diagram to show you the anatomy of the veins. Now the venous sinuses. These are the specialized flattened veins with extremely thin walls. Example, coronary sinus of the heart and the dural venous sinuses of the brain. A valveless, irregularly shaped venous channel is lined by endothelial cells. This is the venous sinuses, dural venous sinuses, which is present in the dura mater. Now here we can see the comparison of arteries and the veins. As we talk of the direction of flow of blood, in case of artery, it is from the heart to the body. In case of veins, it is back to the heart. Oxygen concentration is very much higher. In case of arteries carry pure blood, that is oxygenated blood, and veins carry less oxygenated blood. The exception for the arteries is the pulmonary artery, which is carrying impure blood. An exception for the veins is the pulmonary veins, which is carrying the pure blood. The pressure is higher in arteries in comparison to the veins. And if we talk about the anatomy, there is thicker outer walls, elastic and muscle fibers in the artery. And in case of veins, the outer walls are very thin, elastic and muscle layers. Then if we talk about the location, the arteries are deeply situated in our body, whereas the veins are also very deeply, but there are some superficial veins are also closer to the skin. And walls are absent in the arteries and they are present in the veins. This is the basic diagram to show about the comparison of lumen and the wall between the artery and the vein. Here we can see the lumen is larger in case of vein and the wall is thicker in case of arteries. This is again the comparison between the artery and the vein on the basis of their lumen and the wall thickness. Now the capillaries. Capillaries are the microscopic vessels that connect arterioles and the venules. Their diameter is between 9 to 12 microns and they are found near every cell in the body, but more extensive in highly active tissues like muscles, liver, kidneys and brain. Walls consist of a single layer of simple squamous epithelial cells that allow the exchange of gases. Precapillary sphincters regulate the blood flow. Function is exchange of nutrients and waste between the blood and the tissue fluid. These are the capillary beds. Here we can see precapillary sphincters, which are made up of smooth muscle fibers. They regulate the blood flow from arteries to the capillary bed. Then types of capillaries. First is the continuous capillaries. These are the endothelial cells form a continuous tube except for intercellular clefts. These are found in brain, lungs, skeletal and smooth muscles. Then second type is fenestrated. The plasma membrane has fenestrations or pores. These are found in your kidneys, villi of stomach, intestine, small intestine, and choroid plexus in brain, endocrine glands. Then we have the sinusoids. These are wider, more winding. They are large fenestration and an incomplete basement membrane over here. Protein and RBCs can pass through them and they are found in red bone marrow, liver, spleen and anterior pituitary. Then come to the next topic, that is anastomosis. Here there is union of two or more arteries supplying the same body region. Blockage of any one of the pathway has no effect. Alternate route of blood flow through an anastomosis is known as collateral circulation. So in the diagram we can observe the anastomosis between the artery and the vein. Now, types of anastomosis. First is arterial anastomosis. It is the communication between the arteries or their branches. Example is superficial palmarage and the deep palmarage in case of hand. Here, the superficial palmarage is a communication between the radial and the underl arteries. Similarly, in case of heart, there is anastomosis, coronary anastomosis between the coronaries. Then second is venous anastomosis. It is the communication between the veins and its tributaries. Example is the dorsal venous arch on the dorsum of hand. Then we have arteriovenous anastomosis. It is the communication between the artery and the vein. It is found in skin of nose, lips and external ear. Then there are some arteries which do not anastomose. These are known as end arteries. Example is central artery of retina. Now come to the applied. First is the blood pressure. Blood pressure is defined as the lateral pressure exerted by the flowing blood on the walls of the arteries. Then we have arteriosclerosis. It is the thickening, hardening and loss of elasticity of the walls of the arteries. This is the aneurysm. It is an excessive localized swelling of the wall of an artery. Then this is the Raynaud's phenomena. Here we can see the fingers become white due to lack of blood flow, then blue as vessels dilate to keep the blood in the tissues, and finally as blood flow returns. So this is the triphasic phenomena. Then varicose veins. The veins that are stretched, dilated and overfilled with blood due to incompetent walls. The causes for these are weak walls or heredity, excessive weight, excessive standing, straining, inadequate exercises, increased intrapelvic pressure during pregnancy, constipation. So this is the varicose vein, is how it appears in varicosity. This is the abnormal tortuosity of the veins. Then we have thrombophlebitis. Thrombo means clot. Phlebitis means inflammation of a vein. So thrombophlebitis occurs when a blood clot causes an inflammation in a vein. Blood clots typically form in the legs or pelvis. Formation of venous clot depends on the presence of at least one of virtuous triad factors like venous stasis, injury to the vessel wall, or hypercoagulable state. So this completes our topic, the general anatomy of the blood vessels.